In this video, we are going to talk about the surface area to volume ratio of cells, which is an important concept that relates to why certain cells are shaped the way that they are and why cells never really get above a certain size. So, cells are tiny, you know that already. And so, why can't a cell be as big as an elephant, say? The reason for that is that as a cell increases in size, the volume grows at a faster rate than the surface area does. So if you think of a cell, kind of imagine it as a perfect sphere, a hypothetical perfectly spherical cell, the volume of the cell would represent the amount of cytoplasm that the cell has. The surface area of the cell would represent the amount of membrane that the cell has, and the cell has to exchange all of its nutrients and waste products across that membrane. So the bigger the cytoplasm gets, the more food you need to take into the cell, the more waste products you need to get rid of from the cell. And if you get a bigger and bigger cytoplasm and a smaller and smaller amount of membrane relative to the cytoplasm, eventually you don't have enough surface area to exchange all of the materials that you need across the membrane of the cell. So there's sort of an upper size limit to how big cells can get, and it is based on the ratio of the surface area of the cell to the volume of the cell. If you look at these formulas here, so you could imagine a cell as like a cube or a sphere, the reason that volume increases more quickly than surface area is that volume is either the radius or the length of the side cubed, while surface area is either the radius or the length of the side squared times another number. So, if I started with a radius or a side of 1, when I get a radius or a side of 2, the surface area is going to go up by 2 squared, which is 4 times, and the volume is going to go up by 2 cubed, which is 8 times. So anyway, you could plug different numbers in there, but what you will see is that volume gets bigger faster, surface area gets bigger more slowly. So the bigger the radius of your cell gets, the worse the surface area to volume ratio gets for that cell. Here is a concrete example of that relationship with some math. So in the first cube, you can see that when the side has a length of 1 centimeter, the surface area is 6 centimeters squared, the volume is 1 centimeter cubed, and so the surface area to volume ratio is 6 to 1. That means for if this was a cell, for every 1 cubic centimeter of a really big cell, for every 1 cubic centimeter of cytoplasm, it would have 6 centimeters of centimeters squared of surface area to exchange, oxygen and stuff like that across. Then if you move up to a cube with a side length of 2 centimeters, well, now your surface area goes up to 24, but your volume goes up to 8. So your surface area to volume ratio is now 3 to 1. So now for every 1 centimeter cubed of giant cell cytoplasm, you only have 3 centimeters squared of membrane to exchange materials over. And then if you look at the biggest cube here, the 3 centimeter cube, you see that that ratio's gone down even further to 2 to 1. So you have an even smaller amount of membrane, say, to exchange materials over per amount of cytoplasm than you did before. Here's the same thing shown with hypothetical cells. So if this was a giant cell that was 30 micrometers in side length and it was kind of a cube shaped, it would have a surface area of 5,400 squared micrometers squared. But if you divided that great big cell into, 27 little cells that had the same total volume, they would actually have a much bigger surface area. They would have a surface area of 16,200 micrometers squared. So there's an advantage to making cells smaller because you have a better surface area to volume ratio. One other way that cells will deal with this surface area to volume ratio conundrum is instead of dividing into a bunch of tiny cells, the big cell will get a bunch of little finger-like projections that greatly increase the surface area of the cell. So when we were talking about the cytoskeleton, we talked about microvilli being structures that have actin filaments in them, which are the microfilaments, and microvilli help increase the surface area of cells that need to absorb things. So like cells in the intestines, for example. Here is an image of a bunch of epithelial cells side by side. And you can see that the body of the cell is pretty big, but then at the very top of the cell are these little hair-like, finger-like, teeny tiny projections that absorb things. And that, again, increases the surface area of the cell, so it has a higher surface area to volume ratio than it would if it was like more of a perfect rectangular prism. This is an awesome picture of a bunch of microvilli on cells. You can see some other organelles in the cells too, but these are two cells with microvilli facing each other really closely, and all those little fingers are side by side by side by side. You can see how much surface area that is, because if you think of surface area as like the amount of membrane that you need to cover all of those little projections, it's a lot. And so all of the fluid can get down in the cracks in between those little microvilli, and that fluid can be absorbed all the way up and down the sides, and so it just makes for a lot of surface area that the cells can use to exchange materials. This is a view of microvilli looking down from the top. This is in your nasopharynx, which is the upper part of your throat by your nose. So yeah, you can see all of the surface area created by all of those little finger-like projections. That's how they always describe them in textbooks. So that's the phrase I always use. So yeah, so just understand that as cells grow, the surface area to volume ratio shrinks, which puts an upper limit on cell size, but that some cells get around that by having a bunch of little projections that increase their surface area in relation to their volume.